Good evening to one and all. I'm Maman Mishra of class 9G from Delhi Public School, Siliguri, is going to show the model of DNA, that is deoxyribonucleic acid. DNA is made up of three components. First is phosphate, nitrogenous bases, and the deoxyribose sugar. There are four nitrogenous bases: adenine, thymine, guanine, and cytosine. The adenine combines with thymine with two hydrogen bondings and can be braked easily, whereas in other case, guanine combines with cytosine with three hydrogen bondings and it is very difficult to break them. This is the pic for DNA, that is a double helical structure. Deoxyribonucleic acid is a group of molecule that is responsible for carrying genetic instructions from parents to offsprings. DNA is a nucleic acid present in cells as well as viruses. Viruses in the case I have put here because viruses lie in the borderline of living and non-living. So viruses, when they enter a host living cell, they capture the DNA mechanism property and they make copying of viruses and thus they infect every and each part of the body. DNA are present in chromosomes binded with special kind of proteins that are called histones, and there are also other type of proteins that are dihistones. DNA was proposed in 1953 by biophysicist James D. Watson and Francis Crick. This is the animation for DNA, the DNA, the double helical structure. Moving on to the structure of DNA. DNA is a nucleic acid that is made up of double helix. Double helix meaning that it is made up of two helix with a phosphate. DNA is made up of repeating monomers called nucleotides. Polymers are those which are made up of more than two monomers. They are actually a group of monomers. And DNA is a polymer. It's a biomolecule. And this when this nucleotides, the components, the monomers of DNA, when they come together, they come and they form a repeating unit. That repeating unit is made up of DNA, that is forming DNA molecule, biological terms. These building blocks are made up of three parts: a phosphate group, a sugar, that is deoxyribose sugar, and one of four types of nitrogenous bases, that is adenine, thymine, guanine, and cytosine. These are the parts of the nucleosomes made up of H1 histone. Then we have the octamer and the DNA that is surrounding the octamer. These are the parts of the DNA that I have discussed previously. You can see here it's the base pairing are there. This is a law for base pairing I have explained, and it is having a phosphate backbone as well as the deoxyribose sugar. Law for bases pairing. In this law, I have explained adenine combines with thymine and guanine combines with cytosine. Guanine, when they combine with thymine, it forms two nitrogenous bases, and when they are melted, they can melt easily. Whereas when guanine combines with cytosine, it's having three hydrogen bondings, and thus they can withstand more higher melting points. Inheritance. The four types of nitrogenous bases found in the nucleotides are adenine, thymine, guanine, and cytosine. The order, the sequence of these bases determines what biological instructions are contained in the strand of deoxyribonucleic acid. For example, sequence of ATCGTT, that is adenine, thymine, cytosine, guanine, and thymine might instruct for blue eyes, and while ATCGCT, that is adenine, thymine, cytosine, guanine, and cytosine, thymine, might instruct from brown eyes. These are actually the codes for DNA. Did you know the complete DNA instruction book or genome for a human contains about 3 billion bases and about 20,000 genes on 23 pairs of chromosomes. DNA contains the instructions needed for an organism to develop, survive, and reproduce. To carry out these functions, DNA sequences must be converted into messages that can be used to produce proteins, which are the complex molecules that do the most of the work in the living body. Genes. Genes are the functional working unit of DNA. Each DNA sequence that contains instructions to make a protein is known as a gene. The size of a gene may vary greatly, ranging from about 1000 bases to 1 million bases in humans. Genes only make up about 1% of the deoxyribonucleic acid, that is DNA, sequence. DNA sequences outside this 1% are involved in regulating when, how, and how much a protein is made. Moving on, how are DNA sequences used to make proteins? DNA's instructions are used to make proteins in a two-step process. First, enzymes read the information in a DNA molecule and transcribe into an intermediary molecule called messenger ribonucleic acid or mRNA. Next, the information contained in the mRNA molecule is translated into language of amino acids, that is the simplest form of any protein, which are the building blocks of proteins. This language tells the cell's protein making machinery the precise order in which to link the amino acids produce a specific protein. This is a major task because there are 20 types of amino acids which can be placed in many different orders to form a wide variety of proteins. Central dogma, DNA to RNA to protein. These are the two processes, transcription and reverse transcription. And there's also process of translation. DNA's replication. DNA's unique structure enables the molecule to copy itself during cell division. When a cell prepares to divide, the DNA helix splits down the middle and becomes two single strands. These single strands serve as templates for building two new double-stranded DNA molecules, each a replica of the original DNA molecule. In this process, an base is added whenever there are a T and C, where there is a G, and so until all the bases once again have partners. In addition, when the proteins are being made, the double helix unwinds to allow a single strand of DNA to serve a template. This template strand is the transcript into mRNA, which is a molecule that conveys vital instructions to the cell's protein making machinery. The cell's protein making machinery is basically inside the nucleolus, that is the nucleus of the nucleus. It's a main site of ribosome production site. From there only the ribosomes are being formed. Not directly, but the genetic material of this ribosome is being made over there. DNA replication, this is the method. Moving on, there's one video. Let's watch it. This is how the DNA replication is happening. You can see here. Moving on, next part. What is genetic engineering? Genetic engineering refers to the direct manipulation of DNA to alter an organism's characteristics, the phenotype, in a particular way. This may mean changing one base, that is A to T or C to G, deleting a whole region of DNA, or introducing an additional copy of a gene. Like when the sequence is changed, the mutation that happens over there, and this, like, there's a whole new DNA being formed. The code is different. Like we have seen the genetic codes, the DNA codes for the brown eyes and the blue eyes, etc. The previous slides. Now, why genetic engineering is done? The genetic engineering is used by scientists to enhance or modify the characteristics of an individual organism. Note, this process can also be applied to viruses. Like the pandemic has happened, the scientists have just they have seen the mutations that are in the virus, the COVID virus, SARS-CoV-2, severe acute respiratory syndrome, coronavirus 2, that was also present in the form of MERS. And they just seen this mutation that are happening in the virus, and thus they've prepared different types of vaccination that are present today. So the genetic engineering is very important in this era. This process can also be done by a plasmid. Plasmid is actually a small segment of DNA that are found suspended in the nucleotide region of this DNA. In the cytoplasm, it's suspended nucleoid region. And the mutation is the change in the DNA sequence of an organism. Mutations can result from errors in DNA replication during cell division, exposure to mutagens, and viral infections. Germline mutations that occurs in eggs and sperm can be passed on to offsprings, while somatic mutations that occur in the body cells are being passed on. This credit goes to Aman Mishra for science exhibition. Hope you like this presentation. Thank you.